Molecular evolution and phylogenetics. Molecular evolution refers to the process by which genetic material changes over time. It encompasses the study of changes in gene sequences, gene structure, and their relationship to the evolution of species. Phylogenetics, on the other hand, is the study of the evolutionary relationships among species based on molecular data, e.g., DNA, RNA, or protein sequences. Together, molecular evolution and phylogenetics form the foundation for understanding the genetic basis of evolution and how different species are related through common ancestry. Molecular evolution. Overview. Molecular evolution can be broken down into several core processes. Mutation. Mutations are the fundamental source of genetic variation. They are random changes in DNA sequences and can be caused by various factors, including errors during DNA replication, environmental factors e.g., radiation, chemicals, or viral insertions. Mutations can be classified as point mutations, single nucleotide changes e.g., substitutions, insertions, deletions. Frameshift mutations, insertions or deletions that shift the reading frame of a gene. Silent mutations, mutations that do not affect the protein product due to redundancy in the genetic code. Missense mutations, mutations that change one amino acid in the protein sequence. Nonsense mutations, mutations that result in a premature stop codon. Gene duplication. Gene duplication occurs when a segment of the genome is copied, creating additional copies of a gene. Over time, these duplicate genes may evolve new functions, neofunctionalization, or become non-functional pseudogenization. Gene duplication is a key mechanism in molecular evolution, allowing the accumulation of genetic variation that can lead to the development of novel traits. Genetic drift. Genetic drift is the random fluctuation in allele frequencies due to chance events, particularly in small populations. Over time, genetic drift can lead to the loss or fixation of alleles. Gene flow. Gene flow, or migration, occurs when individuals from different populations interbreed, introducing new genetic material into the population and contributing to genetic variation. Selection. Natural selection acts on genetic variation by favoring advantageous traits and selecting against harmful ones. It leads to the gradual adaptation of populations to their environment. Horizontal gene transfer, HGT. HGT is the transfer of genetic material between organisms that are not parent and offspring, which is common in prokaryotes e.g., bacteria. HGT plays an important role in microbial evolution and can lead to the spread of beneficial traits e.g., antibiotic resistance. Molecular clock hypothesis. The molecular clock hypothesis suggests that mutations accumulate at a relatively constant rate over time, allowing the use of molecular data e.g., DNA sequences to estimate the time of divergence between species. This concept is based on the assumption that genetic mutations occur randomly and are not influenced by selective pressures. Rate of mutation. The rate at which mutations accumulate is typically species-specific, with some genes evolving faster than others. For example, mitochondrial DNA evolves faster than nuclear DNA. Applications. Molecular clocks are widely used in phylogenetics to estimate divergence times between species or populations, aiding in the construction of phylogenetic trees. Phylogenetics. Overview. Phylogenetics is the study of the evolutionary relationships among species or groups of organisms. It is based on the principle of common ancestry, where species are more closely related if they share a recent common ancestor. Phylogenetic trees. A phylogenetic tree is a diagram that represents the evolutionary history of a group of species. It shows how species are related based on shared ancestry and evolutionary events. Phylogenetic trees are built using molecular data, such as DNA or protein sequences, morphological traits, or a combination of both. The branches of the tree represent evolutionary lineages, and the nodes represent common ancestors. Types of phylogenetic trees. Rooted trees. A rooted tree has a single root that represents the most recent common ancestor of all the species in the tree. Unrooted trees. An unrooted tree shows the relationships among species but does not specify the common ancestor. Cladograms. A specific type of phylogenetic tree that shows the branching pattern of species based on shared derived characteristics. Methods of phylogenetic analysis. Maximum likelihood (ML), a statistical method that estimates the tree topology, branching structure, and model of evolution that best explain the observed data. It selects the tree that has the highest probability of producing the observed sequence data. Neighbor joining (NJ), a distance-based method that constructs a tree by iteratively joining pairs of taxa with the smallest genetic distance. Bayesian inference, a probabilistic method that uses Bayesian statistics to infer phylogenetic trees, incorporating prior knowledge and statistical models. Parsimony, the principle that the simplest tree with the fewest evolutionary changes is the most likely. This method is based on the assumption that the most likely phylogeny requires the fewest changes in character states. Molecular markers used in phylogenetics. Molecular markers are genetic sequences used to infer evolutionary relationships. They provide insight into the genetic variation within and between species. DNA sequencing. DNA sequencing is the primary tool used for phylogenetic analysis. By comparing the nucleotide sequences of genes or entire genomes, scientists can reconstruct the evolutionary relationships between species. Mitochondrial DNA, mtDNA. Mitochondrial DNA is inherited maternally and is widely used in phylogenetic studies because it evolves relatively quickly. It is often used to study species that have recently diverged. Ribosomal RNA, rRNA. rRNA genes are highly conserved and are commonly used in phylogenetic analysis to study distant evolutionary relationships. The 16S-rRNA gene is frequently used in prokaryotic phylogenetics, while the 18S-rRNA gene is used for eukaryotes. Single nucleotide polymorphisms, SNPs. SNPs are variations in a single nucleotide-based pair and are widely used as genetic markers in phylogenetic studies. SNPs can be compared across genomes to determine evolutionary relationships. Microsatellites. Microsatellites are short, repetitive DNA sequences that are highly variable among individuals. They are often used to assess genetic diversity within populations. Applications of molecular evolution and phylogenetics. Understanding evolutionary relationships. Phylogenetics helps in understanding the evolutionary relationships among species. For example, it has been used to construct the tree of life, showing how all living organisms are related to each other. Molecular epidemiology. Molecular evolution is used in epidemiology to track the spread of infectious diseases. By analyzing the genetic sequences of pathogens e.g., viruses, bacteria, scientists can trace the source and spread of disease outbreaks. Conservation biology. Phylogenetic analysis can aid in the conservation of endangered species by identifying genetically distinct populations that may require special conservation efforts. Biotechnology. Molecular evolution plays a significant role in the development of biotechnological products, such as genetically modified organisms, GMOs, pharmaceuticals, and enzymes used in industrial processes. Human evolution. Molecular evolution has provided insight into human evolution by comparing human genetic sequences with those of other primates, revealing key differences and similarities. Practice questions and answers. Question 1, GATE 2019. Which of the following molecular markers is most commonly used in phylogenetic studies of prokaryotes? A. 16-srRNA gene. B. Mitochondrial DNA. C. Cytochrome C oxidase gene. D. 18-srRNA gene. Answer. The correct answer is a 16-srRNA gene. The 16-srRNA gene is widely used in prokaryotic phylogenetics because it is highly conserved among bacterial species but still contains enough variability to distinguish between different species. Question 2, GATE 2021. The molecular clock hypothesis suggests that A. Mutations occur at a constant rate over time. B. Mutation rates are constant only for non-coding regions. C. Mutations occur faster in prokaryotes than in eukaryotes. D. Mutations are influenced by natural selection. Answer. The correct answer is that mutations occur at a constant rate over time. The molecular clock hypothesis assumes that genetic mutations accumulate at a roughly constant rate, providing a method for estimating the time of divergence between species. Summary. Molecular evolution and phylogenetics are powerful tools for understanding the evolutionary processes that shape the diversity of life. Through the study of genetic sequences and the construction of phylogenetic trees, scientists can trace the evolutionary history of species, estimate divergence times, and explore the genetic mechanisms underlying evolutionary change. These fields have numerous applications in evolutionary biology, conservation, epidemiology, and biotechnology, making them essential components of modern biological research. You said, neutral theory. ChatGPT said, ChatGPT. Neutral theory of molecular evolution. The neutral theory of molecular evolution, proposed by Moto Kimura in the 1960s, revolutionized the field of molecular evolution by challenging the widely accepted view that natural selection was the primary force driving evolution. According to the neutral theory, most of the genetic variation observed at the molecular level is due to genetic drift rather than selection. In other words, genetic changes are mostly neutral, neither advantageous nor detrimental, and their spread within a population occurs due to random fluctuations in allele frequencies, not because they provide a selective advantage. Key concepts of neutral theory. Neutral mutations. The theory posits that many mutations in an organism's genome are neutral, meaning they do not significantly affect the fitness of the organism. These mutations could be silent mutations which do not change the protein sequence, or mutations that occur in non-coding regions of the genome. Silent mutations may not have any functional consequences but still accumulate in populations over time. Genetic drift. Neutral theory emphasizes genetic drift as the main force behind the fixation or loss of neutral alleles in a population. Genetic drift is a random process, where alleles fluctuate in frequency due to chance events, particularly in small populations. In larger populations, genetic drift has a smaller effect, while in smaller populations, it can lead to the rapid fixation or loss of alleles. Molecular clock. One of the most important implications of the neutral theory is the concept of the molecular clock. Since most mutations are neutral and accumulate at a relatively constant rate over time, the neutral theory provides a means of estimating the time of divergence between species by comparing molecular sequences. Molecular clocks use the rate of accumulation of neutral mutations to estimate how long it has been since two species shared a common ancestor. Hitchhiking. Genetic hitchhiking refers to the phenomenon where a neutral allele's frequency increases or decreases in tandem with a nearby beneficial allele due to linkage disequilibrium. While the neutral allele may not provide any fitness advantage, it hitches a ride with the beneficial allele through natural selection. Substitution rate. According to the neutral theory, the rate of molecular substitution, the rate at which mutations become fixed in a population, is equal to the mutation rate. This means that the rate at which genetic changes accumulate in a population at the molecular level is determined by the rate of neutral mutations rather than by selective pressures. The two types of evolution in neutral theory. Selective evolution. This type of evolution is driven by natural selection, where advantageous traits increase an organism's fitness, leading to the increase in frequency of the associated alleles. In this model, beneficial mutations are preserved and spread in the population due to their positive effect on fitness. Neutral evolution. In contrast to selective evolution, neutral evolution is driven by genetic drift rather than natural selection. In this model, mutations that have no effect on fitness, neutral mutations, spread randomly through populations. Over time, neutral alleles may become fixed or lost purely due to chance. Neutral theory versus Darwinism. The neutral theory differs from Darwinian evolution, which emphasizes natural selection as the primary force driving evolutionary change. While natural selection acts on advantageous traits, the neutral theory posits that most molecular evolution is caused by random genetic drift acting on neutral mutations. This leads to the idea that most genetic changes at the molecular level are not subject to natural selection but are instead driven by random processes. However, it's important to note that both mechanisms, neutral drift and natural selection, are considered to play important roles in evolution, with the neutral theory accounting for a significant portion of molecular evolution, particularly at the level of silent mutations or in non-coding regions of the genome. Evidence supporting the neutral theory. Comparative DNA sequences. One of the key pieces of evidence for the neutral theory comes from comparative studies of DNA sequences across different species. Studies have shown that the rate of molecular evolution in certain genes, e.g., mitochondrial genes, is relatively constant over time and can be used as a molecular clock to estimate the time of divergence between species. This observation is consistent with the idea that mutations accumulate at a roughly constant rate, as predicted by the neutral theory. Silent versus replacement mutations. Silent mutations, those that do not change the protein, accumulate at a faster rate than replacement mutations, those that change the protein. This pattern is consistent with the neutral theory, which suggests that most genetic variation occurs in non-coding regions or in parts of the genome where mutations do not affect fitness. Polymorphisms and fixation. Population genetic studies have found that many genetic polymorphisms, variations within a population, are neutral or nearly neutral and are maintained through genetic drift rather than natural selection. Additionally, many alleles that eventually become fixed, i.e., present in 100% of individuals in a population, do so through random genetic drift rather than through selective advantage. Criticism of the neutral theory. Despite its widespread acceptance, the neutral theory has faced some criticism. Role of natural selection. Some critics argue that the neutral theory underestimates the role of natural selection in molecular evolution, particularly for non-neutral mutations that may have subtle effects on fitness. Recent research suggests that natural selection might act more frequently than the neutral theory predicts, especially in the evolution of protein-coding genes and functional elements of the genome. Environmental adaptations. Neutral theory focuses primarily on neutral mutations and genetic drift, but it does not fully account for adaptive evolution, where mutations provide a fitness advantage and are subject to natural selection. Pseudogenes. Some argue that pseudogenes, non-functional gene copies, and other non-coding regions in the genome, which are often cited as examples of neutral evolution, may still be influenced by selection pressures in certain circumstances, e.g., when they affect gene regulation. Applications of neutral theory. Molecular clock. Neutral theory provides a framework for using molecular data to estimate divergence times between species, the concept known as the molecular clock. By analyzing genetic distances between species and assuming a constant mutation rate, scientists can estimate when species diverge from a common ancestor. Conservation biology. Understanding the molecular evolution of populations can help in the conservation of endangered species. By studying the genetic diversity within populations, conservationists can make more informed decisions regarding breeding programs and conservation strategies. Phylogenetics. Neutral theory helps in interpreting molecular phylogenetic trees, particularly when dealing with gene sequences that evolve under neutral selection, allowing scientists to infer evolutionary relationships based on molecular data. Population genetics. Neutral theory is foundational in understanding genetic drift and how genetic diversity is maintained or lost in small populations, impacting the field of population genetics. Practice questions and answers. Question 1, Gate 2018. Which of the following best describes the primary mechanism of genetic evolution according to the neutral theory? A. Natural selection. B. Genetic drift. C. Gene flow. D. Mutation. Answer. The correct answer is B. Genetic drift. Neutral theory emphasizes genetic drift as the primary mechanism behind the fixation of neutral mutations, rather than selection. Question 2, Gate 2020. The molecular clock hypothesis, as proposed by neutral theory, assumes that A. Mutations accumulate at a constant rate across all species. B. The rate of mutation is proportional to the size of the population. C. Natural selection is the primary driver of molecular evolution. D. Mutations accumulate randomly in non-coding regions of the genome. Answer. The correct answer is that mutations accumulate at a constant rate across all species. The molecular clock hypothesis is based on the idea that mutations accumulate at a relatively constant rate over time, and this can be used to estimate the divergence times between species. Summary. The neutral theory of molecular evolution has had a profound impact on our understanding of molecular evolution. By proposing that most genetic variation at the molecular level is neutral and fixed through genetic drift rather than natural selection, the theory has provided valuable insights into the processes that shape the genetic diversity of populations. It has also paved the way for the development of the molecular clock, a key tool in phylogenetic studies and evolutionary biology. Despite criticisms, neutral theory continues to be an essential framework for understanding the molecular basis of evolution. You said, molecular clocks. ChatGPT said, ChatGPT. Molecular clocks. A molecular clock is a concept in evolutionary biology that uses the rate of genetic mutations to estimate the time of divergence between species or populations. This concept is based on the assumption that mutations accumulate in the genome at a relatively constant rate over time. By comparing the genetic differences between two or more species, scientists can estimate when these species diverge from their common ancestor, assuming that the mutation rate is constant. The idea of molecular clocks is a powerful tool in evolutionary biology, as it allows researchers to construct phylogenetic trees and estimate divergence times without relying solely on fossil records. Molecular clocks are primarily applied to DNA or protein sequence data, as these molecular markers can be used to quantify genetic differences. Key principles of molecular clocks: constant mutation rate. Molecular clocks assume that mutations accumulate at a roughly constant rate over time in a particular lineage. This rate is often referred to as the molecular clock rate. The molecular clock rate can vary between species, genes, and regions of the genome, but the general principle is that neutral mutations, mutations that do not affect an organism's fitness, accumulate steadily and can be used to track evolutionary time. Neutral mutations. The molecular clock concept is primarily based on neutral mutations, mutations that do not affect the fitness of an organism, as they are assumed to accumulate without being influenced by natural selection. Silent mutations in non-coding regions are synonymous mutations, mutations that do not change the amino acid sequence, are often used as markers for molecular clocks because they are less likely to be affected by natural selection. Rate of mutation. The rate of molecular evolution is measured by the substitution rate, which is the rate at which mutations become fixed, i.e., are passed down through generations in a population. The substitution rate can be influenced by factors such as the size of the population, the mutation rate, and the effectiveness of natural selection in removing deleterious mutations. Calibration of the molecular clock. To estimate divergence times using molecular clocks, it is necessary to calibrate the clock using fossil evidence or geological data. This provides an anchor point for the molecular clock by linking genetic differences to actual time. For example, if the fossil record shows that two species diverged 10 million years ago, researchers can use this information to calibrate the molecular clock and estimate divergence times for other species. Genetic data. Molecular clocks typically rely on comparing DNA or protein sequences to measure genetic differences. Common molecular markers used for constructing molecular clocks include mitochondrial DNA, especially in animals, and ribosomal RNA, rRNA genes, both of which evolve at relatively constant rates. Amino acid sequences can also be used, although protein evolution tends to be slower than DNA evolution because of the additional constraints on protein function. Types of molecular clocks. Strict molecular clock. A strict molecular clock assumes that mutations accumulate at a perfectly constant rate across all species and genes. This model is relatively simplistic and does not account for the fact that mutation rates can vary across species or genes. However, it can still be useful for some purposes, especially when comparing closely related species with similar mutation rates. Relaxed molecular clock. A flexible approach is the relaxed molecular clock, which assumes that the mutation rate can vary among different lineages. This model accounts for the fact that mutation rates can differ between species, populations, or even genes within the same genome. The relaxed clock allows researchers to incorporate variations in mutation rates into their estimates of divergence times. Autonomous molecular clock. Some molecular clocks, such as those used for mitochondrial DNA, evolve more independently of the nuclear genome and can serve as an autonomous molecular clock. These types of clocks are particularly useful in reconstructing evolutionary histories and estimating divergence times. Applications of molecular clocks. Dating the divergence of species. The primary application of molecular clocks is to estimate the time of divergence between species or populations that do not have fossil records. By comparing the genetic differences between species and calibrating the molecular clock with fossil or geological data, scientists can estimate when the species last shared a common ancestor. Molecular clocks are particularly useful in cases where fossils are scarce or difficult to interpret. Constructing phylogenetic trees. Molecular clocks are used to construct phylogenetic trees by estimating the genetic distances between species or populations. The molecular clock approach is particularly valuable when reconstructing evolutionary relationships between species that have diverged in the distant past and for which fossil data is unavailable. Tracking the evolution of genes. Molecular clocks can be used to study the evolution of specific genes or gene families. By measuring the rate of mutation in a gene across different species, scientists can gain insights into the functional constraints on that gene and how it has evolved over time. For example, studying the evolution of genes involved in immune response or developmental processes can provide important insights into the adaptive evolution of these genes. Conservation biology. Molecular clocks can be used in conservation biology to estimate the genetic diversity and evolutionary history of endangered species. By comparing the genetic diversity of closely related species, researchers can gain insights into the population history of endangered species and make more informed conservation decisions. Virus evolution. Molecular clocks are widely used to study the evolution of viruses, especially RNA viruses, which mutate rapidly. By applying molecular clocks to viral genomes, scientists can trace the spread of viral outbreaks and estimate when they originated. For example, molecular clocks have been used to study the evolution of the HIV virus and to track the emergence and spread of new influenza strains. Criticism and limitations of molecular clocks. Rate variation. One of the major criticisms of molecular clocks is that mutation rates are not constant across all species or genes. The rate of evolution can vary due to factors such as population size, environmental pressures, and differences in DNA repair mechanisms. This variation in mutation rates can lead to inaccuracies in estimating divergence times if the clock is not calibrated properly. Selective pressure. While molecular clocks rely on neutral mutations, some mutations may be subject to natural selection. Mutations that confer a selective advantage, beneficial mutations, may accumulate at a faster rate than neutral mutations, which can distort the molecular clock. This can be particularly problematic when studying genes or regions of the genome that are under strong selective pressure, such as protein-coding genes involved in immune response or adaptation to environmental changes. Lineage-specific rates. Some species or lineages may evolve more rapidly or slowly than others, making it difficult to apply a uniform molecular clock rate across all species. Relaxed molecular clocks attempt to address this issue by allowing for variation in mutation rates between different lineages. Calibration issues. The accuracy of molecular clock estimates depends on the availability and reliability of calibration points, such as fossil records or geological events. If calibration points are inaccurate or uncertain, the molecular clock estimates can be misleading. Practice questions and answers. Question 1. Date 2019. The molecular clock hypothesis assumes that A. Mutations accumulate at a constant rate across all species. B. The rate of mutation is proportional to the size of the population. C. Natural selection is the primary driver of molecular evolution. D. Mutations accumulate randomly in non-coding regions of the genome. Answer. The correct answer is that mutations accumulate at a constant rate across all species. The molecular clock hypothesis assumes that mutations accumulate in a linear, constant manner over time, providing a way to estimate divergence times between species. Question 2. Date 2020. In molecular clock theory, which of the following best describes the assumption about mutation rates? A. The mutation rate is constant only in protein-coding regions. B. The mutation rate is constant across all genes and species. C. The mutation rate is constant across all species but varies in different genes. D. The mutation rate is influenced only by natural selection. Answer. The correct answer is C. The mutation rate is constant across all species but varies in different genes. While the mutation rate is assumed to be constant over time for a given gene or region, it can vary across different genes or species due to different evolutionary pressures. Summary. The molecular clock is a valuable tool for understanding the timing of evolutionary events, based on the premise that genetic mutations accumulate at a relatively constant rate. It provides insights into the divergence times of species, even in the absence of fossil records. Despite its utility, the molecular clock is subject to certain criticisms, including rate variation across species and the influence of natural selection on mutation rates. To mitigate these issues, scientists use a relaxed molecular clock that accounts for lineage-specific mutation rates. Overall, the molecular clock remains an important technique for studying evolutionary history and constructing phylogenetic relationships. You said, rates of evolution. ChatGPT said, ChatGPT. Rates of evolution. The rate of evolution refers to the speed at which genetic changes accumulate in a population over time, leading to changes in traits or the emergence of new species. Evolution can occur at different rates depending on various factors, including the type of genetic change, e.g., mutations, genetic drift, natural selection, the environment, and the reproductive strategies of organisms. Understanding the rates of evolution helps to explain the patterns and processes observed in the evolution of species and populations. Factors influencing the rates of evolution. Several factors influence the rate at which evolution occurs, including the following. 1. Mutation rate. Mutations are the ultimate source of genetic variation, and the mutation rate is a key factor in determining the rate of evolution. A higher mutation rate generally leads to faster evolution because new genetic variants, alleles, are introduced more quickly into the gene pool. Mutations can be neutral, beneficial, or harmful. Neutral mutations are those that do not affect the fitness of the organism, while beneficial mutations may increase the organism's survival or reproductive success, and harmful mutations may reduce fitness. Mutation rate can vary across different genes and species. For example, RNA viruses typically have much higher mutation rates than DNA-based organisms, leading to faster evolutionary changes in viral populations. 2. Genetic drift. Genetic drift refers to the random changes in allele frequencies in a population due to chance events, especially in small populations. The effect of genetic drift is more pronounced in small populations, where alleles may become fixed, reach a frequency of 100%, or lost, reach a frequency of 0% by chance. In larger populations, genetic drift has less of an effect on allele frequencies, as selection and other factors tend to have a stronger influence. However, in small populations or during bottleneck events, sharp reductions in population size, drift can significantly impact the rate of evolution. 3. Natural selection. Natural selection is one of the most important forces driving the rate of evolution. It acts on phenotypic variation within a population, favoring individuals with traits that enhance survival and reproduction, while eliminating those with less advantageous traits. Selection pressures, such as predation, competition for resources, and climate change, can accelerate the rate of evolution by strongly favoring certain alleles over others. When a trait is under strong positive selection, e.g., resistance to disease, it can spread rapidly through a population, leading to faster evolutionary change. On the other hand, negative selection, or purifying selection, which removes harmful alleles, can slow down the rate of evolution by maintaining the stability of the genome and preventing the accumulation of detrimental mutations. 4. Gene flow, migration. Gene flow, or migration, refers to the movement of individuals and their genes between different populations. This process can introduce new genetic material into a population, which may speed up the rate of evolution, especially if the migrating individuals bring beneficial traits. Gene flow can reduce genetic differences between populations, potentially slowing down the process of speciation. However, in populations that are isolated and experience little gene flow, the rate of evolution can be slower due to a lack of genetic diversity. 5. Population size. The effective population size, which is the number of individuals in a population contributing genetically to the next generation, plays a significant role in the rate of evolution. In smaller populations, genetic drift is more pronounced, and evolutionary changes can occur more rapidly through random fluctuations in allele frequencies. Larger populations generally experience more stable evolutionary processes because natural selection and gene flow are more likely to dominate, and genetic drift has a weaker influence. 6. Reproductive strategy. The reproductive strategy of a species can affect the rate of evolution. Species with short generation times, such as bacteria or insects, can evolve more rapidly because they reproduce more frequently, allowing more opportunities for genetic mutations to occur and spread through the population. In contrast, species with long lifespans and longer generation times, such as elephants or humans, tend to evolve more slowly because each generation is spaced farther apart, and the accumulation of genetic changes takes longer. 7. Environmental change. Rapid environmental changes, such as shifts in climate, habitat destruction, or changes in available resources, can increase the rate of evolution by imposing strong selective pressures on populations. In response to environmental challenges, organisms may undergo rapid adaptations, e.g., evolving resistance to a new disease or adapting to new food sources, which accelerates evolutionary change. Extinction events, such as asteroid impacts or volcanic eruptions, can also drive rapid evolutionary changes by drastically altering environmental conditions and leading to the extinction of some species while providing new ecological niches for others to evolve into. Types of evolutionary rates. Gradualism. Gradualism refers to the idea that evolution occurs slowly and steadily over long periods of time. This model suggests that species evolve through the accumulation of small, incremental changes in genetic traits. Gradual changes in populations are often driven by small mutations, genetic drift, and natural selection acting over many generations. The punctuated equilibrium hypothesis, proposed by Stephen Jay Gould and Niles Eldredge, challenges the gradualist view, suggesting that evolution may occur in rapid bursts followed by long periods of stasis, no significant change. Punctuated equilibrium. Punctuated equilibrium proposes that most evolutionary change happens in relatively brief periods of rapid change, followed by long periods of little or no change. This contrasts with the gradualism model, which sees evolutionary change as slow and constant. This pattern may occur due to environmental shifts, such as climate changes or ecological upheaval, which can create opportunities for rapid adaptation or speciation. Adaptive radiation. Adaptive radiation refers to the rapid diversification of a single ancestral species into a variety of forms that occupy different ecological niches. This phenomenon typically occurs when a population colonizes a new environment with diverse available resources, leading to the evolution of many new species in a relatively short period. The classic example of adaptive radiation is the evolution of the Darwin's finches in the Galapagos Islands, where different species evolved from a common ancestor to exploit different food sources and ecological niches. Measuring the rate of evolution. Molecular evolution. The rate of molecular evolution can be measured by comparing genetic sequences between species. By quantifying the number of mutations or substitutions that occur in a gene or genome over a given period, scientists can estimate the rate of molecular evolution. Molecular clocks described earlier are often used to estimate the rate of molecular evolution by assuming a constant mutation rate over time. Fossil record. The fossil record provides another way to measure the rate of evolution by looking at the changes in morphological traits of species over time. The rate of morphological change can be inferred from the fossil record, although this method may be limited by gaps in the fossil record or the difficulty of interpreting certain evolutionary trends. Practice questions and answers. Question 1, GATE 2018. Which of the following factors would likely increase the rate of evolution in a population? A. Decrease in population size. B. High mutation rate. C. Lack of natural selection. D. Low reproductive rate. Answer. The correct answer is B. High mutation rate. A high mutation rate increases genetic variation within a population, which can accelerate the process of evolution, especially if beneficial mutations increase in frequency through natural selection. Question 2, GATE 2020. What is the primary difference between gradualism and punctuated equilibrium in terms of evolutionary rate? A. Gradualism suggests rapid evolutionary change, while punctuated equilibrium suggests slow change. B. Punctuated equilibrium suggests rapid evolutionary change, with long periods of stasis, while gradualism suggests slow and continuous change. C. Gradualism and punctuated equilibrium both suggest rapid change. D. Gradualism and punctuated equilibrium both suggest no change over time. Answer. The correct answer is B. Punctuated equilibrium suggests rapid evolutionary change, with long periods of stasis, while gradualism suggests slow and continuous change. Punctuated equilibrium proposes that evolutionary change happens in bursts, whereas gradualism posits that change occurs steadily over time. Summary. The rate of evolution is influenced by several factors, including mutation rate, genetic drift, natural selection, population size, reproductive strategy, and environmental conditions. Evolution can occur at varying rates, from slow and gradual changes, gradualism, to rapid bursts of change followed by periods of stasis, punctuated equilibrium. Understanding the factors that influence evolutionary rates allows researchers to better explain the patterns of evolution observed in nature and provides insights into how species adapt to changing environments. You said. Phylogenetic reconstruction, ChatGPT said. ChatGPT. Phylogenetic reconstruction. Phylogenetic reconstruction is the process of determining the evolutionary relationships between different species or groups of organisms based on molecular, morphological, or other types of data. The goal of phylogenetic reconstruction is to create a phylogenetic tree, also known as a tree of life, which visually represents the evolutionary history and relatedness of organisms. Phylogenetic trees depict the divergence of species from common ancestors and can be used to infer the sequence of evolutionary events. Key concepts in phylogenetic reconstruction. Phylogenetic tree. A phylogenetic tree is a branching diagram that represents the evolutionary relationships among species or other taxa. Each branch point or node in the tree represents the common ancestor of the taxa that diverge from that point. The tips of the branches represent the species or groups under study, and the length of the branches may reflect the degree of genetic divergence or evolutionary time. Cladogram. A cladogram is a simplified version of a phylogenetic tree that shows the relationships between species based on shared derived characteristics, synapomorphies. Cladograms do not show the timescale of evolutionary changes or the amount of genetic divergence, but they illustrate the branching pattern of evolution. Homology and homoplasy. Homology refers to traits or genes that are inherited from a common ancestor. These traits are used to infer evolutionary relationships because they reflect shared ancestry. Homoplasy, on the other hand, refers to traits that evolve independently in different lineages due to convergent evolution or evolutionary reversals, not because of common ancestor. Homoplastic traits can complicate phylogenetic reconstruction because they may mislead researchers into drawing incorrect evolutionary relationships. Molecular data versus morphological data. Molecular data, such as DNA, RNA, or protein sequences, is increasingly used for phylogenetic reconstruction because it provides a wealth of genetic information. Molecular data can be more reliable than morphological data, as it is less subject to convergence or reversal. Morphological data, such as physical traits e.g., body structure, skeletal features, leaf shape, have traditionally been used in phylogenetic studies, especially when molecular data is unavailable. However, morphological data can sometimes lead to indirect conclusions if convergent traits are mistaken for homologies. Outgroup comparison. An outgroup is a species or group of species that is related to, but not part of, the group being studied, the ingroup. The outgroup is used to determine the direction of character evolution. Traits that are present in both the outgroup and ingroup were considered plesiomorphies, ancestral traits, while traits that are only present in the ingroup were considered apomorphies, derived traits. Methods of phylogenetic reconstruction. Several methods can be used to reconstruct phylogenies, ranging from traditional approaches based on morphological traits to more recent molecular methods. Cladistics. Cladistics is a method of phylogenetic reconstruction that classifies species based on shared derived characteristics, synapomorphies. Cladistics aims to group organisms that share common evolutionary ancestry. It uses a process called character coding to score traits in a matrix, where species are listed along the rows and characters' traits are listed along the columns. The data matrix is then analyzed using algorithms, e.g., parsimony analysis, to generate the most likely tree, where the simplest tree with the fewest evolutionary changes, least number of steps, is preferred. Maximum parsimony. Maximum parsimony (MP) is a method of tree construction that aims to find the tree that requires the least number of evolutionary changes to explain the observed data. In other words, it chooses a tree that minimizes the total number of character state changes, e.g., mutations, across all characters. MP is a simple and intuitive approach, but it can be computationally expensive for large datasets, and it may not always produce the most accurate tree if the data is complex. Maximum likelihood (ML). Maximum likelihood is a more sophisticated method that uses statistical models to estimate the probability of a particular phylogenetic tree given the observed data. It takes into account the likelihood of different character state changes, e.g., nucleotide substitutions, occurring over time. ML methods are computationally intensive but can provide more accurate trees, especially when working with large molecular datasets or complex evolutionary models. Bayesian inference. Bayesian inference is a probabilistic approach to phylogenetic reconstruction that uses Bayes' theorem to estimate the probability of different phylogenetic trees. It incorporates prior knowledge about the evolutionary process and updates the probability of a tree based on the observed data. Bayesian methods use Markov chain Monte Carlo (MCMC) algorithms to sample from a space of possible trees and estimate the posterior probability of each tree. This method provides a measure of confidence posterior probability for each branch in the tree. Neighbor joining (NJ). Neighbor joining is a distance-based method that constructs a phylogenetic tree by calculating pairwise genetic distances between taxa and then progressively clustering the closest taxa into groups. It is fast and widely used for large datasets, although it may not always produce the most accurate trees if the evolutionary processes are complex. Steps in phylogenetic reconstruction. Data collection. The first step in phylogenetic reconstruction is to gather data on the species or taxa being studied. This may include molecular data, e.g., DNA, RNA, or protein sequences, or morphological traits. The choice of data depends on the available resources and the evolutionary questions being addressed. Character selection and alignment. If molecular data is used, the next step is to align the sequences of the taxa being studied to ensure that homologous positions derived from a common ancestor, are compared. This step is critical to ensure that the analysis is comparing equivalent sites across the species. For morphological data, the researcher must identify and code the relevant traits, e.g., the presence or absence of a specific feature, in a matrix. Tree construction. Once the data is collected and aligned, the next step is to apply one of the phylogenetic reconstruction methods, e.g., maximum parsimony, maximum likelihood, to generate a phylogenetic tree. The tree is usually constructed by analyzing the data matrix and applying algorithms to identify the most likely evolutionary relationships. Tree evaluation. After constructing the phylogenetic tree, it is important to assess the reliability of the tree using methods such as bootstrap resampling or posterior probabilities in Bayesian methods. This helps to evaluate the confidence in the inferred evolutionary relationships. The tree can also be compared to known evolutionary data, such as fossil records or other phylogenies, to validate its accuracy. Practice questions and answers. Question 1, Gate 2015. Which of the following methods of phylogenetic reconstruction is based on the principle of minimizing the number of evolutionary changes? A. Maximum likelihood B. Maximum parsimony C. Neighbor joining D. Bayesian inference answer. The correct answer is B. Maximum parsimony. Maximum parsimony is a tree-building method that selects the tree requiring the fewest number of evolutionary changes to explain the observed data. Question 2, Gate 2017. In the context of phylogenetic reconstruction, what is the role of an outgroup? A. It is used to identify homoplastic traits in the ingroup. B. It helps in identifying shared derived traits in the ingroup. C. It is used to root the phylogenetic tree and infer the direction of character evolution. D. It provides genetic material for comparison with the ingroup. Answer. The correct answer is C. It is used to root the phylogenetic. Cladistics aims to group organisms that share common evolutionary ancestry. It uses a process called character coding to score traits in a matrix, where species are listed along the rows and characters' traits are listed along the columns. The data matrix is then analyzed using algorithms, e.g., parsimony analysis, to generate the most likely tree, where the simplest tree with the fewest evolutionary changes, least number of steps, is preferred. Maximum parsimony. Maximum parsimony (MP) is a method of tree construction that aims to find the tree that requires the least number of evolutionary changes to explain the observed data. In other words, it chooses a tree that minimizes the total number of character state changes, e.g., mutations, across all characters. MP is a simple and intuitive approach, but it can be computationally expensive for large datasets, and it may not always produce the most accurate tree if the data is complex. Maximum likelihood (ML). Maximum likelihood is a more sophisticated method that uses statistical models to estimate the probability of a particular phylogenetic tree given the observed data. It takes into account the likelihood of different character state changes, e.g., nucleotide substitutions, occurring over time. ML methods are computationally intensive but can provide more accurate trees, especially when working with large molecular datasets or complex evolutionary models. Bayesian inference. Bayesian inference is a probabilistic approach to phylogenetic reconstruction that uses Bayes' theorem to estimate the probability of different phylogenetic trees. It incorporates prior knowledge about the evolutionary process and updates the probability of a tree based on the observed data. Bayesian methods use Markov chain Monte Carlo (MCMC) algorithms to sample from a space of possible trees and estimate the posterior probability of each tree. This method provides a measure of confidence posterior probability for each branch in the tree. Neighbor joining (NJ). Neighbor joining is a distance-based method that constructs a phylogenetic tree by calculating pairwise genetic distances between taxa and then progressively clustering the closest taxa into groups. It is fast and widely used for large datasets, although it may not always produce the most accurate trees if the evolutionary processes are complex. Steps in phylogenetic reconstruction. Data collection. The first step in phylogenetic reconstruction is to gather data on the species or taxa being studied. This may include molecular data, e.g., DNA, RNA, or protein sequences or morphological traits. The choice of data depends on the available resources and the evolutionary questions being addressed. Character selection and alignment. If molecular data is used, the next step is to align the sequences of the taxa being studied to ensure that homologous positions derived from a common ancestor are compared. This step is critical to ensure that the analysis is comparing equivalent sites across the species. For morphological data, the researcher must identify and code the relevant traits, e.g., the presence or absence of a specific feature in a matrix. Tree construction. Once the data is collected and aligned, the next step is to apply one of the phylogenetic reconstruction methods, e.g., maximum parsimony, maximum likelihood, to generate a phylogenetic tree. The tree is usually constructed by analyzing the data matrix and applying algorithms to identify the most likely evolutionary relationships. Tree evaluation. After constructing the phylogenetic tree, it is important to assess the reliability of the tree using methods such as bootstrap resampling or posterior probabilities in Bayesian methods. This helps to evaluate the confidence in the inferred evolutionary relationships. The tree can also be compared to known evolutionary data, such as fossil records or other phylogenies, to validate its accuracy. Practice questions and answers. Question 1, Gate 2015. Which of the following methods of phylogenetic reconstruction is based on the principle of minimizing the number of evolutionary changes? A. Maximum likelihood. B. Maximum parsimony. C. Neighbor joining. D. Bayesian inference. Answer. The correct answer is B. Maximum parsimony. Maximum parsimony is a tree-building method that selects the tree requiring the fewest number of evolutionary changes to explain the observed data. Question 2, Gate 2017. In the context of phylogenetic reconstruction, what is the role of an outgroup? A. It is used to identify homoplastic traits in the in-group. B. It helps in identifying shared derived traits in the in-group. Cladistics aims to group organisms that share common evolutionary ancestry. It uses a process called character coding to score traits in a matrix, where species are listed along the rows and characters' traits are listed along the columns. The data matrix is then analyzed using algorithms, e.g., parsimony analysis, to generate the most likely tree, where the simplest tree with the fewest evolutionary changes, least number of steps, is preferred. Maximum parsimony. Maximum parsimony (MP) is a method of tree construction that aims to find the tree that requires the least number of evolutionary changes to explain the observed data. In other words, it chooses a tree that minimizes the total number of character state changes, e.g., mutations, across all characters. MP is a simple and intuitive approach, but it can be computationally expensive for large datasets, and it may not always produce the most accurate tree if the data is complex. Maximum likelihood (ML). Maximum likelihood is a more sophisticated method that uses statistical models to estimate the probability of a particular phylogenetic tree given the observed data. It takes into account the maximum parsimony (MP) is a method of tree construction that aims to find the tree that requires the least number of evolutionary changes to explain the observed data. In other words, it chooses a tree that minimizes the total number of character state changes, e.g., mutations, across all characters. MP is a simple and intuitive approach, but it can be computationally expensive for large datasets, and it may not always produce the most accurate tree if the data is complex. Maximum likelihood (ML). Maximum likelihood is a more sophisticated method that uses statistical models to estimate the probability of a particular phylogenetic tree given the observed data. It takes into account the likelihood of different character state changes, e.g., nucleotide substitutions, occurring over time. ML methods are computationally intensive but can provide more accurate trees, especially when working with large molecular datasets or complex evolutionary models. Bayesian inference. Bayesian inference is a probabilistic approach to phylogenetic reconstruction that uses Bayes' theorem to estimate the probability of different phylogenetic trees. It incorporates prior knowledge about the evolutionary process and updates the probability of a tree based on the observed data. Bayesian methods use Markov chain Monte Carlo (MCMC) algorithms to sample from a space of possible trees and estimate the posterior probability of each tree. This method provides a measure of confidence posterior probability for each branch in the tree. Neighbor joining (NJ). Neighbor joining is a distance-based method that constructs a phylogenetic tree by calculating pairwise genetic distances between taxa and then progressively clustering the closest taxa into groups. It is fast and widely used for large datasets, although it may not always produce the most accurate trees if the evolutionary processes are complex. Steps in phylogenetic reconstruction. Data collection. The first step in phylogenetic reconstruction is to gather data on the species or taxa being studied. This may include molecular data, e.g., DNA, RNA, or protein sequences or morphological traits. The choice of data depends on the available resources and the evolutionary questions being addressed. Character selection and alignment. If molecular data is used, the next step is to align the sequences of the taxa being studied to ensure that homologous positions derived from a common ancestor are compared. This step is critical to ensure that the analysis is comparing equivalent sites across the species. For morphological data, the researcher must identify and code the relevant traits, e.g., the presence or absence of a specific feature in a matrix. Tree construction. Once the data is collected and aligned, the next step is to apply one of the phylogenetic reconstruction methods, e.g., maximum parsimony, maximum likelihood, to generate a phylogenetic tree. The tree is usually constructed by analyzing the data matrix and applying algorithms to identify the most likely evolutionary relationships. Tree evaluation. After constructing the phylogenetic tree, it is important to assess the reliability of the tree using methods such as bootstrap resampling or posterior probabilities in Bayesian methods. This helps to evaluate the confidence in the inferred evolutionary relationships. The tree can also be compared to known evolutionary data, such as fossil records or other phylogenies, to validate its accuracy. Practice questions and answers. Question 1, Gate 2015. Which of the following methods of phylogenetic reconstruction is based on the principle of minimizing the number of evolutionary changes? A. Maximum likelihood B. Maximum parsimony C. Neighbor joining D. Bayesian inference answer. The correct answer is B. Maximum parsimony. Maximum parsimony is a tree-building method that selects the tree requiring the fewest number of evolutionary changes to explain the observed data. Question 2, Gate 2017. In the context of phylogenetic reconstruction, what is the role of an outgroup? A. It is used to identify homoplastic traits in the in-group. B. It helps in identifying shared derived traits in the in-group. C. It is used to root the phylogenetic tree and infer the direction of character evolution. D. It provides the genetic material for comparison with the in-group. Answer. The correct answer is C. It is used to root the phylogenetic tree and infer the direction of character evolution. An outgroup is used in phylogenetic reconstruction to determine the ancestral versus derived states of characters and to root the tree. Summary. Phylogenetic reconstruction is a crucial method for understanding. The correct answer is C. It is used to root the phylogenetic tree and infer the direction of character evolution. An outgroup is used in phylogenetic reconstruction to determine the ancestral versus derived states of characters and to root the tree. Summary. Phylogenetic reconstruction is a crucial method for understanding the evolutionary relationships between species. By using molecular, morphological, and other data, scientists can build phylogenetic trees that depict the branching patterns of evolution. Several methods, such as cladistics, maximum parsimony, maximum likelihood, and Bayesian inference, are employed to reconstruct these trees. Each method has its advantages and limitations, and the choice of method depends on the type and size of data, as well as the evolutionary questions being addressed. Phylogenetic reconstruction is essential for studying the diversity of life and the processes that drive evolutionary change. You said. Molecular systematics. ChatGPT said. ChatGPT. Molecular systematics. Molecular systematics is the use of molecular data, primarily DNA, RNA, and protein sequences, to infer the evolutionary relationships between species or groups of organisms. This approach complements traditional methods of classification based on morphology and has revolutionized the study of biodiversity, providing more accurate insights into evolutionary processes. Molecular systematics uses molecular markers to trace genetic divergence, reconstruct phylogenetic trees, and provide a more reliable method of classifying organisms. Key concepts in molecular systematics. Molecular markers. Molecular markers are specific sequences of nucleotides, DNA or RNA, or amino acids, proteins, that can be used for comparison across different species or populations. The most commonly used molecular markers in systematics are mitochondrial DNA, mtDNA. This is a common marker because it is inherited maternally and has a high mutation rate, making it useful for studying population-level diversity and phylogenetic relationships. Nuclear DNA. These markers are inherited from both parents and can be used to study deeper evolutionary relationships. Chloroplast DNA, cpDNA, often used in plant systematics because of its high conservation and maternal inheritance in most plants. Ribosomal RNA, rRNA, particularly the small subunit ribosomal RNA, ssURRNA, is commonly used in higher taxonomic levels, as it is highly conserved across species. Single nucleotide polymorphisms, SNPs, these are variations at a single nucleotide position in the genome and are useful for high-resolution genetic studies. Microsatellites. These are short, repetitive sequences found in both coding and non-coding regions of the genome. Genetic divergence. Molecular systematics relies on measuring genetic divergence to infer evolutionary relationships. Organisms that are more genetically similar are believed to be more closely related. This divergence can be measured by comparing the genetic differences between species or populations using sequence alignment and distance methods. Phylogenetic trees. Phylogenetic trees or gene trees are constructed to represent the evolutionary relationships of species or populations based on molecular data. These trees can be constructed using various methods, such as neighbor joining, NJ, a distance-based method that clusters taxa based on genetic distances. Maximum likelihood, ML, a probabilistic method that estimates the most likely tree given a model of molecular evolution. Bayesian inference, a statistical method that uses probability theory to estimate phylogenetic trees, taking into account prior distributions and sampling variations. Parsimony, the simplest tree, requiring the fewest number of evolutionary changes, is considered the best. Homology versus analogy. In molecular systematics, it is crucial to distinguish between homologous and analogous traits. Homology refers to traits or sequences inherited from a common ancestor, whereas analogy refers to traits that are similar due to convergent evolution and not due to shared ancestry. Misidentifying analogous traits as homologous can lead to incorrect phylogenetic conclusions. Molecular clocks. Molecular clocks are used in molecular systematics to estimate the timing of evolutionary events. The basic assumption is that genetic mutations accumulate at a roughly constant rate over time, so the number of mutations between two species can be used to estimate the time since they last shared a common ancestor. However, mutation rates can vary between species, so molecular clocks are calibrated using fossil data or known geological events. Steps in molecular systematics. Data collection. The first step in molecular systematics is the collection of genetic material. This may include the extraction of DNA, RNA, or protein sequences from the organisms of interest. The DNA can be extracted from tissue samples, blood, or even preserved specimens. Once the genetic material is collected, specific genes or regions are selected for sequencing based on their suitability for resolving the phylogenetic question, e.g., rRNA genes for deep evolutionary relationships, or mitochondrial genes for population-level studies. Sequence alignment. After sequencing, the next step is to align the DNA or protein sequences from different species or populations to identify homologous positions. This is often done using bioinformatics tools such as ClustalW, MAFFT, or Muscle. Sequence alignment is critical because it ensures that the comparisons are made between equivalent regions of the genomes, avoiding erroneous conclusions. Phylogenetic tree construction. Once the sequences are aligned, the next step is to construct a phylogenetic tree using various methods, such as neighbor joining, maximum likelihood, or Bayesian inference. The tree reflects the evolutionary relationships between the species based on their molecular differences. Trees can be rooted using an outgroup, which is a taxon that is closely related to the in-group but not part of it, to help determine the direction of evolutionary changes. Tree evaluation. After constructing the phylogenetic tree, it is important to evaluate the robustness of the tree. Bootstrap analysis is commonly used to assess the support for different branches in the tree by resampling the data and creating multiple trees to test the stability of the results. Posterior probabilities and Bayesian methods or consensus trees from multiple iterations of tree building can also help assess the reliability of the inferred relationships. Interpretation and analysis. The final step is to interpret the phylogenetic tree in the context of evolutionary history. The tree can provide insights into the divergence times of species, the patterns of speciation, and the evolutionary processes that have shaped biodiversity. Molecular systematics can also be used to infer evolutionary processes such as gene flow, mutation rates, and selection in populations, helping to provide a more complete understanding of evolutionary dynamics. Applications of molecular systematics. Species identification and classification. Molecular systematics has greatly enhanced the ability to identify species and classify organisms. DNA barcoding, for example, uses short genetic markers, often from mitochondrial DNA, to rapidly identify species and assess biodiversity, even in cryptic or morphologically similar species. Evolutionary relationships. Molecular systematics is used to resolve the evolutionary relationships among species, genera, families, and higher taxonomic levels. It has helped clarify many difficult phylogenies that were ambiguous based on morphology alone. Biogeography. Molecular data are used to trace the geographic distribution of genetic diversity and infer historical processes such as migration, isolation, and speciation. For example, molecular systematics can help determine whether current species distributions are the result of ancient dispersal events or more recent colonization. Conservation biology. Molecular systematics is used in conservation biology to assess genetic diversity within populations, identify evolutionary significant units (ESUs), and guide conservation strategies. It helps in understanding the genetic health of endangered species and determining the degree of population structure in fragmented habitats. Understanding evolutionary processes. Molecular systematics provides a detailed view of evolutionary processes, including mutation, genetic drift, natural selection, and gene flow. By comparing genetic data from different taxa, researchers can infer the relative contributions of these processes in shaping genetic diversity. Practice questions and answers. Question 1, GATE 2016. Which of the following molecular markers is most commonly used in plant systematics due to its maternal inheritance and lack of recombination? A. Nuclear DNA B, mitochondrial DNA C, chloroplast DNA D, ribosomal RNA. Answer. The correct answer is C, chloroplast DNA. Chloroplast DNA is inherited maternally in most plants and is frequently used in plant systematics due to its relatively high mutation rate and lack of recombination. Question 2, GATE 2014. In molecular systematics, what is the primary purpose of using an outgroup when constructing a phylogenetic tree? A. To root the tree and determine the ancestral state of characters. B. To estimate the divergence time between species. C. To identify homologous sequences across species. D. To validate the alignment of sequences. Answer. The correct answer is A. To root the tree and determine the ancestral state of characters. An outgroup is used to root the phylogenetic tree and infer the direction of character evolution, ensuring that ancestral and derived traits are correctly identified. Summary. Molecular systematics is a powerful tool for studying evolutionary relationships by using molecular markers to build phylogenetic trees. It provides a more accurate and reliable method of classifying organisms and understanding evolutionary processes than traditional morphological methods alone. By using molecular data such as DNA, RNA, and protein sequences, researchers can uncover hidden biodiversity, trace evolutionary history, and study processes like gene flow and speciation. The application of molecular systematics is essential in fields such as evolutionary biology, conservation, and systematics. You said, macroevolution. ChatGPT said, ChatGPT macroevolution. Macroevolution refers to large-scale evolutionary changes that occur above the level of species, leading to the formation of new taxonomic groups such as families, orders, classes, and phyla. Unlike microevolution, which deals with small-scale changes within a population, like changes in allele frequencies, macroevolution is concerned with significant evolutionary events that shape the diversity of life over long periods of geological time. It encompasses speciation, adaptive radiation, extinction events, and the emergence of novel traits and body plans. Macroevolutionary processes can be observed as the accumulation of microevolutionary changes over long periods, but the distinction between macro and microevolution is not always clear-cut. The processes that drive both are essentially the same, but the scope and timescale of macroevolution are much broader. Key concepts in macroevolution. Speciation. Speciation is the process by which one species splits into two or more distinct species. It is a central feature of macroevolution and is responsible for the generation of biodiversity. There are several types of speciation. Allopatric speciation occurs when a population is geographically isolated, leading to reproductive isolation and divergence into separate species. Sympatric speciation occurs when new species arise within the same geographic area, often due to ecological, behavioral, or genetic differences. Parapatric speciation happens when populations are adjacent but not fully geographically isolated, leading to speciation due to limited gene flow and different selection pressures across the range. Parapatric speciation involves a small population that is isolated at the periphery of a larger population, leading to speciation due to genetic drift and founder effects. Adaptive radiation. Adaptive radiation is a process in which a single ancestral species rapidly diversifies into a wide variety of forms that adapt to different ecological niches. It often occurs when new habitats become available, such as after the extinction of dominant groups or when organisms colonize new environments. Classic examples of adaptive radiation include the evolution of the finches on the Galapagos Islands and the diversification of mammals after the extinction of dinosaurs. Mass extinctions. Mass extinctions are large-scale events in which a significant proportion of Earth's species become extinct within a relatively short period of time. These events create opportunities for surviving groups to diversify and fill the ecological niches left vacant by extinct species. Major mass extinctions have occurred throughout Earth's history, such as the end Permian and end Cretaceous events, which radically reshaped the course of evolution. After mass extinctions, biotic recovery often leads to evolutionary radiation, where surviving species rapidly diversify. Macroevolutionary patterns. Convergent evolution. Different species evolve similar traits independently, often because they occupy similar ecological niches. For example, wings evolved in both bats and birds, despite their different evolutionary lineages. Divergent evolution. A single ancestral species gives rise to different species with distinct adaptations. This can lead to the formation of new genera, families, and even orders. Parallel evolution. Closely related species evolve in a similar manner due to similar selective pressures. This is seen in species that occupy similar niches in different geographic locations. Coevolution. This is when two or more species evolve in response to each other's adaptations. For example, the coevolution of flowering plants and their pollinators is a key aspect of macroevolutionary processes. Evolution of novel traits. The emergence of novel traits, such as wings in insects and birds or the evolution of photosynthesis in plants, can drive macroevolution by enabling organisms to exploit new ecological niches. These traits often arise through mutations in key genes and their modification through natural selection. The evolution of key innovations, such as the development of the amniotic egg in reptiles, allows for the colonization of new environments and contributes to the diversification of life forms. Punctuated equilibrium. Punctuated equilibrium is a theory proposed by Stephen Jay Gould and Niles Eldredge, suggesting that evolutionary change occurs in rapid bursts, separated by long periods of stasis, little or no change. This contrasts with the traditional view of gradualism, which proposes that evolution is a slow and steady process. Punctuated equilibrium may occur due to sudden environmental changes, genetic mutations, or the opening of new ecological niches. The fossil record and macroevolution. The fossil record provides critical evidence for macroevolution, offering insights into past biodiversity, extinction events, and major transitions in the history of life. Fossils of extinct species help scientists understand evolutionary trends and patterns, such as the development of complex body plans and the emergence of major groups of organisms. The Cambrian explosion is a classic example of macroevolution, where a rapid diversification of life forms occurred approximately 541 million years ago, leading to the appearance of most major animal phyla in the fossil record. Theories of macroevolutionary change. Gradualism. Gradualism is the idea that evolutionary change occurs gradually over long periods of time, with small incremental changes accumulating in populations. This model was popularized by Charles Darwin and suggests that the slow accumulation of small genetic variations can lead to the formation of new species over time. Punctuated equilibrium. As mentioned earlier, punctuated equilibrium proposes that most species exhibit little evolutionary change for most of their geological history, with rapid bursts of speciation occurring during periods of environmental change or ecological opportunity. Saltationism. Saltationism is the theory that large evolutionary changes, called saltations, can occur in a single generation due to mutations in key developmental genes, leading to dramatic phenotypic changes. Although this theory has largely fallen out of favor, some researchers argue that large mutations can indeed play a role in major evolutionary transitions. Evolutionary developmental biology, Evo-Devo. Evo-Devo studies the role of developmental processes in evolution. It focuses on how changes in the regulation of genes during development can lead to the evolution of new body plans and structures. For instance, mutations in the Hox genes, which control the development of body segments, can lead to major morphological changes. Examples of macroevolution. Evolution of mammals. Mammals evolved from reptilian ancestors during the Mesozoic era. The diversification of mammals after the extinction of dinosaurs about 66 million years ago is an example of adaptive radiation, where mammals evolved to fill ecological roles vacated by the dinosaurs. The evolution of flowering plants. Flowering plants, angiosperms, emerged during the Cretaceous period, evolving from seed plants. Their diversification involved the development of unique structures like flowers, fruits, and seeds, which enabled them to dominate terrestrial ecosystems. Cetacean evolution. Cetaceans, whales, dolphins, and porpoises evolved from terrestrial mammals. Fossil evidence shows the transition from land-dwelling mammals to fully aquatic animals, with changes in limb structure, body size, and the development of specialized features like echolocation. Practice questions and answers. Question 1, GATE 2019, which of the following is not a characteristic of punctuated equilibrium? A. Evolution occurs in rapid bursts. B. Evolution occurs slowly and gradually. C. Species exhibit stasis for long periods. D. Major evolutionary changes occur during environmental shifts. Answer. The correct answer is B. Evolution occurs slowly and gradually. Punctuated equilibrium suggests that evolution occurs in rapid bursts followed by long periods of stasis, contrasting with the gradualism model. Question 2, GATE 2017, the phenomenon in which a single ancestral species diversifies rapidly into a variety of forms adapted to different ecological niches is known as A. Convergent evolution. B. Divergent evolution. C. Adaptive radiation. D. Parallel evolution. Answer. The correct answer is C. Adaptive radiation. Adaptive radiation refers to the rapid diversification of an ancestral species into multiple forms that occupy different ecological niches. Summary. Macroevolution involves large-scale evolutionary changes that result in the emergence of new species, higher taxonomic groups, and novel biological traits. It includes processes like speciation, adaptive radiation, mass extinctions, and the evolution of new body plans. Understanding macroevolution provides insights into the long-term patterns of life on Earth and is critical for studying the origins of biodiversity. Macroevolutionary processes operate over vast geological timescales, shaping the complexity and diversity of life as we know it today. You said, species concepts. ChatGPT said, ChatGPT. Species concepts. A species is traditionally defined as a group of organisms that can interbreed and produce fertile offspring, but this concept has evolved over time to incorporate a range of perspectives. Various species concepts have been proposed to address the complexity of speciation, the processes through which new species emerge, and the variety of ways in which species can be classified based on different criteria. Here, we explore the most prominent species concepts, their strengths and weaknesses, and their application in modern evolutionary biology. 1. Biological species concept BSC. The biological species concept was introduced by Ernst Mayr in 1942 and is one of the most widely recognized species concepts. It defines species as groups of actually or potentially interbreeding populations that are reproductively isolated from other such groups. Key features. Species are reproductively isolated from one another, meaning they cannot produce fertile offspring if they interbreed. Reproductive isolation can be prezygotic before fertilization or postzygotic after fertilization. Focuses on gene flow within populations and the lack of gene flow between species. Strengths. Provides a clear criterion for identifying species in terms of reproductive isolation. Widely applicable in sexually reproducing organisms. Weaknesses. Does not apply to asexual organisms, e.g., bacteria, some fungi. Cannot be used to classify extinct species of those known only from fossils. Difficult to test in nature due to the challenges of determining reproductive isolation in all cases. Example. Eastern and western meadowlarks are two species of birds that are morphologically very similar but do not interbreed because they have different songs and mating behaviors. 2. Morphological species concept MSC. The morphological species concept is one of the oldest species concepts, proposed by Linnaeus. It defines species based on shared morphological physical traits, often relying on structural characteristics to distinguish species. Key features. Species are defined based on morphological similarity and distinct physical features. It is often used in the classification of fossils and organisms that do not reproduce sexually. Strengths. Can be applied to fossils and asexual organisms. Provides a practical approach for identifying species in the absence of genetic or reproductive data. Weaknesses. Phenotypic plasticity can lead to misidentification. Different environments may cause organisms to appear differently. May not account for cryptic species that look morphologically similar but are genetically distinct. Interspecific variation within a species may lead to errors in distinguishing species. Example. Dogs and wolves are morphologically similar but are considered different species based on their genetic divergence and reproductive isolation, though they can interbreed under certain circumstances. 3. Phylogenetic species concept PSC. The phylogenetic species concept defines a species as the smallest group of individuals that share a common ancestor and can be distinguished by unique genetic or morphological characteristics. Key features. Species are identified based on a monophyletic group, a group that includes a common ancestor and all of its descendants. Utilizes genetic data, e.g., DNA sequences, to define species and identify differences between populations. Strengths. Can be applied to both sexual and asexual organisms. Useful for identifying species in evolutionary terms, emphasizing common ancestry. Can be used to classify extinct species based on genetic data extracted from fossils, e.g., through DNA sequencing. Weaknesses. Requires comprehensive genetic data, which may not be available for all organisms. Can lead to the recognition of very fine distinctions between populations, which may not always be meaningful or practical for conservation purposes. Example. Phylogenetic analysis of cichlid fish in African lakes has revealed a large number of closely related species that are morphologically similar but genetically distinct. 4. Ecological species concept, ESC. The ecological species concept, proposed by Van Balen in 1976, defines a species as a set of organisms that occupy a unique ecological niche. It emphasizes the importance of ecological factors in maintaining species boundaries. Key features. Species are defined by their niche, the role they play in their environment, including interactions with other organisms and abiotic factors. Considers the ability of species to adapt to specific environments and resources. Strengths. Can be applied to both sexual and asexual organisms. Useful in understanding ecological interactions and adaptive radiations. Allows for species definitions that reflect their ecological role rather than just reproductive isolation. Weaknesses. The concept of an ecological niche can be difficult to define in some cases. It may overlap with other species that occupy similar ecological niches, leading to ambiguity. Example. Two species of Darwin's finches, Geospiza, may be considered separate species if they occupy different ecological niches, e.g., one feeding on large seeds and the other on small seeds, even though they might be able to interbreed. 5. Genotypic cluster species concept. This concept defines a species based on clusters of genetic similarity. If populations are genetically distinct, they are considered separate species. Key features. Species are identified based on a monophyletic group, a group that includes a common ancestor and all of its descendants. Utilizes genetic data, e.g., DNA sequences, to define species and identify differences between populations. Strengths. Can be applied to both sexual and asexual organisms. Useful for identifying species in evolutionary terms, emphasizing common ancestry. Can be used to classify extinct species based on genetic data extracted from fossils, e.g., through DNA sequencing. Weaknesses. Requires comprehensive genetic data, which may not be available for all organisms. Can lead to the recognition of very fine distinctions between populations, which may not always be meaningful or practical for conservation purposes. Example. Phylogenetic analysis of cichlid fish in African lakes has revealed a large number of closely related species that are morphologically similar but genetically distinct. 4. Ecological species concept, ESC. The ecological species concept, proposed by Van Balen in 1976, defines a species as a set of organisms that occupy a unique ecological niche. It emphasizes the importance of ecological factors in maintaining species boundaries. Key features. Species are defined by their niche, the role they play in their environment, including interactions with other organisms and abiotic factors. Considers the ability of species to adapt to specific environments and resources. Strengths. Can be applied to both sexual and asexual organisms. Useful in understanding ecological interactions and adaptive radiations. Allows for species definitions that reflect their ecological role rather than just reproductive isolation. Weaknesses. The concept of an ecological niche can be difficult to define in some cases. It may overlap with other species that occupy similar ecological niches, leading to ambiguity. Example. Two species of Darwin's finches, Geospiza, may be considered separate species if they occupy different ecological niches, e.g., one feeding on large seeds and the other on small seeds, even though they might be able to interbreed. 5. Genotypic cluster species concept. This concept defines a species based on clusters of genetic similarity. If populations are genetically distinct, they are considered separate species. Key features. Species are defined by genetic clusters, where there is a significant genetic difference between populations that leads to distinct evolutionary trajectories. Emphasizes genetic divergence over reproductive isolation or ecological factors. Strengths. Useful for identifying cryptic species, species that are genetically distinct but morphologically similar. Can be applied to both sexual and asexual organisms. Can work in the case of gene flow, where reproductive isolation may not yet have fully occurred. Weaknesses. It can be difficult to determine the threshold for genetic divergence that warrants classifying two populations as separate species. Genetic differences may not always correspond to ecological or morphological differences. Example. Cryptic species of frogs that look nearly identical but have significant genetic differences and exhibit different behaviors. 6. Recognition species concept, RSC. The recognition species concept defines a species by its ability to recognize potential mates. According to this concept, species are groups of organisms that can recognize each other as mates through specific signals or behaviors. Key features. Species are defined by mate recognition. It focuses on the behavioral aspects of speciation, particularly in terms of reproduction. Strengths. Provides a behavioral definition of species. Can be applied to both sexual and asexual organisms. Weaknesses. May not be applicable to species with no obvious mating behaviors or recognition signals. Mate recognition may not always be a clear barrier between species, especially in cases where hybridization occurs. Example. Firefly species that use distinct flashing patterns to recognize mates and prevent interbreeding with other species. Comparison of species concepts. Species concept focus best used for limitations. Biological species concept reproductive isolation sexual organisms, especially animals, does not apply to asexual organisms or fossils. Morphological species concept physical characteristics fossils, organisms with similar morphologies may miss cryptic species or oversimplify phenotypic diversity. Phylogenetic species concept genetic relationships sexual and asexual organisms. Evolutionary studies requires genetic data, can lead to fine distinctions. Ecological species concept ecological niche ecologically distinct populations niche definition may be vague or overlapping. Genotypic Cluster species concept genetic divergence cryptic species. Evolutionary genetics genetic divergence thresholds can be difficult to establish recognition species concept mate recognition signals organisms with specific mating behaviors does not apply to species with no obvious mating signals practice questions and answers. Question 1 Gate 2018 Which of the following species concepts is most useful for identifying species in asexual organisms? A. Biological species concept B. Morphological species concept C. Ecological species concept D. Phylogenetic species concept answer. The correct answer is D. Phylogenetic species concept. The phylogenetic species concept is useful for identifying species in asexual organisms because it relies on genetic differences rather than reproductive isolation. Question 2 Gate 2020 According to the biological species concept, what is the primary criterion for determining whether two populations belong to different species? A. Physical differences B. Genetic divergence C. Reproductive isolation D. Ecological niche answer. The correct answer is C. Reproductive isolation. Biological species concept defines species based on the ability of populations to interbreed and produce fertile offspring, with reproductive isolation preventing gene flow between species. Summary. Species concepts are essential frameworks for understanding biological diversity and defining what constitutes a species. Each concept has its strengths and limitations, and the appropriate concept may vary depending on the context of the study and the organisms involved. The most commonly used species concepts include the biological species concept, morphological species concept, phylogenetic species concept, and others, each emphasizing different aspects of species identification, such as reproduction, genetics, morphology, and ecology. Understanding these concepts allows researchers to better classify and study the diversity of life on Earth. You said, and speciation, ChatGPT said, ChatGPT speciation. Speciation is the evolutionary process by which populations of a single species evolve into distinct species. It involves the accumulation of genetic, phenotypic, and behavioral differences that result in reproductive isolation between formerly interbreeding populations. Speciation is a fundamental concept in evolutionary biology, providing insight into the mechanisms that generate biological diversity over time. It occurs when gene flow between populations is interrupted and genetic divergence begins. This process can happen in various ways, leading to different types of speciation depending on the ecological, geographical, and genetic factors involved. Types of speciation. There are two primary mechanisms by which speciation can occur. Allopatric speciation, sympatric speciation. Additionally, there are other processes such as parapatric speciation and parapatric speciation which represent specific variations on these models. 1. Allopatric speciation. Allopatric speciation occurs when a population is geographically divided by a physical barrier, e.g., mountain ranges, rivers, glaciers, or human activities resulting in the isolation of subpopulations. The physical separation prevents gene flow between the groups, allowing them to evolve independently over time. Genetic differences accumulate due to mutations, genetic drift, and selection pressures, leading to reproductive isolation. Key features of allopatric speciation. Geographical isolation is the primary driver. Gene flow is interrupted between subpopulations. Genetic drift and natural selection can act independently on each group. Over time, reproductive barriers develop, e.g., changes in mating behavior, physical differences, or genetic incompatibilities. Example. The Darwin's finches on the Galapagos Islands provide a classic example of allopatric speciation. Different island populations of finches evolved into distinct species due to geographic isolation and different environmental pressures on each island. Strengths of allopatric speciation, well supported by empirical evidence and widely observed in nature. Can lead to significant genetic divergence, providing clear examples of speciation. 2. Sympatric speciation. Sympatric speciation occurs when new species arise from a single population within the same geographic area. Unlike allopatric speciation, there is no physical barrier separating the populations. Instead, speciation can occur due to ecological isolation, where different subgroups of a population exploit different niches or ecological resources. Reproductive isolation may arise through genetic changes, behavioral differences, or temporal isolation, even when individuals live in close proximity. Key features of sympatric speciation. No geographic barrier to gene flow. Occurs in populations that inhabit the same area but become reproductively isolated. Often driven by ecological factors like resource differentiation, mate preference, or temporal isolation. Can be facilitated by polyploidy, doubling of chromosome number, in plants, creating instant reproductive isolation. Example. Cichlids in East African lakes exhibit sympatric speciation, where different species have evolved within the same lake, exploiting different ecological niches like varying food sources or habitats. Apple maggot flies, Rhagoletis pomonella, provide an example where different populations specialize on different host plants, apple versus hawthorn, even though they occupy the same geographic area. Strengths of sympatric speciation. Demonstrates how ecological factors and behavior can drive speciation without physical separation. Can be rapid and lead to high levels of biodiversity in certain environments. Challenges. It's more difficult to demonstrate compared to allopatric speciation due to the lack of a clear physical barrier. The genetic changes that promote sympatric speciation are often subtle and harder to detect. 3. Parapatric speciation. Parapatric speciation occurs when two populations are adjacent to each other and experience limited gene flow. While there is no strict geographic barrier, environmental differences across a range of the populations can promote divergent selection. This leads to reproductive isolation between the populations, especially when hybrids between them have reduced fitness. Key features of parapatric speciation. Adjacent populations with a gradient of environmental conditions. Limited gene flow across the contact zone. Divergent selection due to ecological differences. Reproductive isolation is often due to hybrid disadvantage, i.e., hybrids are less fit than either parent species. Example. Eurasian grasshoppers Strathypus along a cline, where one population is adapted to one type of habitat and the other to a different one, and hybrid grasshoppers show reduced fitness. 4. Parapatric speciation. Parapatric speciation is similar to allopatric speciation but involves smaller populations that are isolated at the periphery of the larger population's range. This model is sometimes called the founder effect model because small, isolated populations may experience genetic drift more strongly, leading to rapid divergence. Key features of parapatric speciation. Small, peripheral populations are isolated from the main population. Genetic drift plays a significant role in divergence due to the small population size. The isolated population is more likely to experience genetic bottlenecks, which can increase the chances of speciation. Example. A population of a bird species that becomes isolated on a small island might undergo parapatric speciation. The small island population may develop distinct characteristics, including changes in morphology or behavior, due to genetic drift and adaptation to the island's environment. Mechanisms of speciation. The mechanisms that contribute to the process of speciation include genetic drift, random changes in allele frequencies in small populations, natural selection, differential survival and reproduction based on environmental pressures, leading to adaptations, mutation, new genetic variants that contribute to divergence. Reproductive isolation, any mechanism that prevents gene flow between populations, which can be prezygotic e.g., temporal isolation, behavioral isolation, or postzygotic e.g., hybrid sterility. Reproductive isolation. Reproductive isolation is the hallmark of speciation and can be categorized into two types. Prezygotic isolation, mechanisms that prevent fertilization from occurring. Temporal isolation, species breed at different times, e.g., different seasons.